I'm Michael. I'm currently a postdoc at Dominic Neopek's group at the IPMB Heidelberg. Before that, I was at Jan Korbel's group at EMBL, where I worked on developing machine learning models for protein generation, protein design. And today I will talk about one of these models, the salad model, and how we used it to efficiently generate proteins for a variety of applications. Right. So let me start maybe, yeah, by saying that Salad is currently open source software. So if you want to give it a try, you can just go to GitHub and it should be fairly easy to install, given you have a Python installation. So, all right. So why design proteins at all? So there's a lot of proteins that have interesting applications in, say, basic biology, but also in industry and in medicine. For example, enzymes like this xylanase here on the left, they're used fairly extensively across industry, this one in particular in the paper making process. And it might be interesting to be able to generate and design custom enzymes for custom applications as they come up. On the right, this antibody heavy and light chain bound to spike RBD is another example of a very useful proteins. In general, antibodies are interesting proteins as therapeutics, but also as markers in immunofluorescence or similar approaches in the lab. So it might be also interesting or useful if we were able to design custom antibodies or in general custom protein binders to any given target we might want to design for. Right. That's also from a more basic science perspective, being able to design proteins of increasing complexity might tell us something about how these complex molecular machines that we see in nature actually work. Right? By building them, we can try understanding what's required to make such complex machines work. All right, so protein design with machine learning. How does this work? So in the last couple of years, starting mostly with the release of AlphaFold a couple of years ago, AlphaFold 2 a couple of years ago, there's been this development of many different tools coming up, machine learning based tools coming up that do certain things in the process of protein design. And one thing there's currently a lot of models for, which is, I guess, one of the central steps of protein design using machine learning, is protein structure generation. So having models where you can feed in a specification of what type of protein you want, and then having the model spit out a protein backbone that is a protein structure without the side chains that follows the specification, right? And for that, there is RSO, there is RF diffusion, there is Chroma, there's Gini2, there's like a million different diffusion models that kind of do this. And then we get those backbones for a specification, and we stick them into a sequence design model, most commonly protein MPNN or ligand MPNN from the Biker lab. And this gives us sequences. So it populates a protein sequence on top of the backbone we previously designed. And then, of course, we need to check if those sequences fit the backbone so we can decide what sequences to order and make in the lab. And for that, currently, mostly we use AlphaFold or ESMFold, depending on the approach, to predict the structure of each of the sequences we get out of protein MPNN, and then compare it with the original design backbone and see, okay, are they self-consistent and are they confidently predicted? And if so, we call a backbone that came out of the structure generator designable. Optionally, we might use Rosetta or other physics-based tools as additional filters for designability, but in most cases, it's always protein MPNN design and then AlphaFold or ESMFold scoring on self-consistency and confidence. So in terms of what actually falls out of AlphaFold or ESMFold, we look at the PLDDT, the predicted local distance difference test for the predicted structure of each of the design sequences. And this is a measure of local quality of the designed backbone or local confidence of the designed backbone. And the higher it is, the more confident AlphaFold is in its prediction. And as a second thing, we look at consistency between the predicted structure and the input design, which we do by computing the RMSD, the root mean square deviation between the two structures. And we call this self-consistent RMSD, SCRMSD. And for ESMFold and for AlphaFold, there is thresholds for both of these that then tell us, okay, everything that falls within those thresholds we call designable. And this is kind of the basis of figuring out in a vacuum without experimental confirmation how good different protein structure generators are compared to each other by comparing the percentage of designs that fall in this designable region. Right, so why yet another model? Before I mentioned that there is many models in protein design, protein structure generation, but all of these models seem to share a set of trade-offs that they do some things well, other things, well, not as well. And these trade-offs kind of range around the quality of generated structures. So how designable are the structures and how well do they fit the specifications we feed in? The flexibility of the method. So what types of different tasks can we solve with the methods without having to try a new model? And finally, the speed of the method. So the throughput we can achieve, how many designs we can churn out per minute. And also what is very important for experimentation here is the time to first design. So how long do we have to wait to see the first structure out of the model to see if we have to change anything? Right. And so most diffusion models like RF diffusion or Genie, they fall somewhere along the quality and speed axis. So where they produce reasonable design abilities at reasonably high speed, but they lack flexibility as they have to be retrained from task to task. For example, if you want to do binder design, you have to train a separate model. If you want to do motif scaffolding, you have to train a separate model and so on. On the other hand, hallucination-based methods like RSO, they give pretty good quality and are very flexible, but they are also very slow. And then there's Chroma, which for some reason seems to be very flexible and very fast, but the designability lags behind comparable models. So we kind of want to be able to improve the models and pipelines here to get somewhere in the middle of this triangle. And also, we need to be able to go beyond data limitations. So there's lots of types of proteins where there is very little data in the PDB, or the data diversity is low. One of these cases is antibodies. There's just not many antibody-antigen complexes in the PDB. And another very pressing case in this case is proteins that change conformation upon some stimulus. And because the PDB is mostly snapshots, mostly crystal structures, which show a single snapshot of a protein, uh, there is very little data on conformational change of proteins that can be extracted from there. So either we need to improve the data situation, or what is easier computationally is we need to somehow creatively force our models to do out of distribution generation. Right. So how did we do this with Salad? How did we improve existing models while not having to train anything too large and staying within the compute budget that is available to a regular researcher without the amount of compute available to a large company? Right. So we started with denoising generative models. So with denoising diffusion models like RF diffusion, which are trained to take noisy structures of proteins together with conditioning information and iteratively turn that into realistic protein structures. And for that, we built Salad around a kind of encoder decoder architecture. So during training, we take protein structures, we encode them with an attention-based encoder that encodes each amino acid as its backbone atoms, so the N, Ca, Co, and C beta atoms, plus an additional cloud of pseudoatoms. So we don't treat the side chains explicitly here, but instead turn them into these pseudoatoms that each amino acid has the same amount of. And then we train the model as a diffusion model by adding noise to these encoded structures and training a denoiser module to remove that noise again, and finally feed the denoise structure into a amino acid decoder with an architecture quite like protein MPNN that takes the output structure, predicts amino acid sequences, and generates side chains for these sequences. And doing this, we use an architecture that is different from previous approaches in that it does not compute interactions between all pairs of amino acids in a protein, but instead has each amino acid only look at its nearest neighbors. First in sequence, so amino acids that are adjacent to each other in the amino acid chain are each other's neighbors. Then in space, so we take an additional set of 16 neighbors in Euclidean space around each amino acid. And then in addition, we also sample randomly additional amino acids which are farther away. So each residue in the protein, each amino acid, kind of sees its immediate neighbors, but also a sample of the whole protein. And this way, we get a model that has a complexity of n, the number of amino acids in a protein, times k, a constant number of neighbors, which greatly reduces compute time relative to models that are n squared in complexity or even n cubed, like RF diffusion. Also, we do not use that many parameters in our models. So in the end, the pure diffusion model that does the generation is just under 9 million parameters, whereas comparable models, previous models, they're larger than that. Right. And using this efficient architecture and small model, we were able to generate proteins, protein monomers across all sizes between 50 amino acids and 1,000 amino acids. So here you see a couple example generations where the design structure is overlaid with the ESM for prediction of its design sequence. As you can see, the errors here, the RMSDs, are fairly low across all of these sizes. If you look at this quantitatively for a number of different models across all of these sizes from 50 to 1,000 amino acids, we can see some interesting things here. So all the gray models and black models in here are previous methods, and the colored models here are different variants of our salad architecture that we continually improved to reach designability, higher designability at larger and larger sizes. So at first, we tried training Salad on the same data and with the same training approach as RF diffusion, and we found that despite being smaller and more efficient, it's not exactly worse than RF diffusion in terms of designability. So the orange curve is the Salad model, and the light gray curve here is RF diffusion. But it displays the same type of behavior that around 500 amino acids, suddenly performance starts dropping, and we get almost no designable structures at 1,000 amino acids. So we wondered why that might be, and changed the way the model gets noisy protein structures, so change the way noise is added to the proteins, and developed this shaped noise approach to noising the input protein structures, which allowed us to generate proteins up to 1,000 amino acids long with reasonable design abilities. And also with high diversity in terms of secondary structure. So looking at the secondary structure distribution for natural proteins in the AlphaFold database, the distribution you get out of RF diffusion and the one you get out of salad on the very right, you can see that uh, whereas RF diffusion seems to be very biased for purely alpha helical structures and nearly produces no pure beta sheet structures over here, salad produces a good mix of everything, all beta, all alpha, and in between. Um, and all of this happens fairly quickly per design. So if you go on a coffee break and you run RF diffusion for 400 amino acid designs, you come back after 10 minutes and you get 5 designs. If you do the same thing and run salad instead, you come back to 158 designs and the same level of designability or higher. So this is around a 31 times speed up over the previous state of the art. Looking at this just in terms of numbers for different models, we can see that our salad models here at the bottom, the pink one and the blue one, need the least time per generation on a consumer GPU across all sizes, even compared to other efficient protein generation models like Chroma here in orange or Proteina from NVIDIA here in black. So yeah, the model seems to be pretty fast. So we have a model. It can do things, but can it do interesting things? So far, we've talked about applications that are pretty much toy problems, just generating a protein that is predicted to fold and that is globular and that is monomeric. But none of this is very useful for actual protein design. So we wanted to see if we could use some tricks during inference, during sampling from the model, to allow Salad to design proteins that are out of distribution relative to its training data set and use that to solve some more interesting problems in protein structure generation. So as a first step towards that, we first wanted to see if we can maximize the diversity of structures we can get out of salad and use that to cover as wide a space of protein designs as possible for all the subsequent tasks we wanted to look at. And for that, we conditioned salad with random secondary structure strings. So in order to get rid of any secondary structure bias, we instead sampled a fraction for alpha helices and beta strands from a uniform distribution, then instantiated secondary structure elements, so helices and strands of that length, and randomly arranged them into a string, so into a sequence of secondary structure annotation for each amino acid, and then conditioned a salad model to generate structures based on this annotation, like these ones here on the right. And doing this randomly, we were able to generate these diverse structures you can see down here. And how diverse are they really? So we did this for 50,000 generated backbones with random secondary structure conditioning. And we found that comparing these backbones to each other, 91.4% of these were unique when clustered at a TM score below 0.6. And they were also mostly designable. So even though we started with random secondary structures, by doing sequence design with protein MPNN and prediction with ESM-fold, we found that 81.4% of these were predicted to fold properly. And of these folded ones, 11,973, so over 10,000, were novel when compared to structures in the PDB, so had a very low TM score compared to structures in the PDB. Also looking at the secondary structure distribution up here, or at the entropy of this distribution directly down here, we can see that doing this random secondary structure conditioning pushes the entropy of the secondary structure distribution across all protein sizes, which translates to us having a more diverse set of secondary structures to work with when generating structures this way. Right. Now, having sorted out designability and diversity for our models, we looked at how we can make them do things they weren't trained for. And for this, we developed a new way of sampling these models, which we call structure editing. So instead of just feeding noise and conditioning information into a model and taking out a structure, we, at each step of the generation, we edited the input noise and the output structure of the model to encode structural constraints. For example, we could use this to generate proteins that were predicted to fold differently as monomers or in a complex. We were able to generate proteins that were predicted to fold in a specific shape, or that scaffolded specific motifs from other proteins, or that were predicted to bind a desired target. So how does this work in a concrete example? So if you want to scaffold a motif from some protein, for example, an enzyme active site, what we could do is we could start with random noise, stick this into our salad model, get out a denoise structure, and then take our motif of interest, align it to the denoise structure, and replace the coordinates in the denoise structure with the coordinates of the motif. And if we did that at every step of our denoising process, we would end up with scaffolded motifs like these, where we ran this for a standard benchmark of motif scaffolding tasks from the RF diffusion and Genie2 pipers. We were able to scaffold most of these motifs there. So out of 30 motifs in the benchmark, we were able to solve 28, which is one more than Genie2, the previous state of the art. As you can see here in those examples, the RMSD between the motif and the design structure is pretty low. So it seems the model is doing something right here. We were also able to use the same approach to encode various types of symmetries. Before Salad's protein design of symmetric proteins with diffusion models was mostly limited to point symmetric proteins. We were able to extend this to proteins with a screw axis symmetry. So where you not only have rotation around an axis, but you have rotation and simultaneous translation around the axis of symmetry. And so we were able to design proteins like these that were predicted to also fold like this, and where extended repeats were also predicted to fold in the same way by AlphaFold 3. So yeah, we were also able to do silly things like shape condition design, having the model spell out its own name. The trick to that is fairly easy. Instead of starting from a point cloud of random noise, instead we initialize random noise around positions in the letter. So if you look at the letter S over here, at each of these positions here, we would initialize a random point cloud of amino acids and force the model to place the amino acids in the denoise structure around that starting point. If we did that at every step in generation, we could generate basically any shape like this. One of the more interesting things we were able to do this way was to do multi-state design. So multi-state design with proteins is basically designing one sequence that folds in two different ways, depending on the conditions. And in this case, the conditions were that we wanted to have one full-length parent sequence with a mixed alpha-beta fold, that when it was split down the middle into two child sequences, which are basically the N terminus and the C terminus of the parent, these child sequences should be predicted to fold as helix bundles, so losing the beta sheet in the middle here. And here's a couple examples of how Salad did on this task, where in colored you see the predicted structure using AlphaFold 2, and in gray you see the original design for the parent and child structures. But it has to be said that this only worked with a really modest success rate of 2.9% of backbones. So only 2.9% of backbones we generated this way out of 1,000 total generated backbones were able to recover this multi-state behavior, where for a whole 16%, we had partial success in the sense that the parent and one of the children were predicted to fold the correct way, and the other child was predicted to fold in the wrong way. So it's really not a good success rate yet. However, compared to the second
Michael,
best mod, yes,
yeah, about multi-step design.
yes.
I'm wondering, maybe I've missed how do you couple these multiple denoising processes to come up with the result, like the process of
Right,
yes.
right. I think I have a backup slide somewhere. If I just go through here, there should be one, or maybe not. Okay, never mind then. So the way this works is the way this works is you run three separate denoising processes, right? One for the parent and one for each of the child structures, right? And then you know from your specification what the secondary structure should be for the parent and what the secondary structure should be for the children, right? And for the parts where we know that the structure of the children is the same as the structure of the parent, then at each step of generation, we align the denoise structure of the child to the denoise structure of the parent and take the average, right? And replicate that average across the two states. So basically, the parts that are the same in secondary structure are kept the same in tertiary structure, while the parts that are different in secondary structure are allowed to change between the three processes.
Wow, sounds novel.
So it's
Yeah,
it's pretty much a hack to make this work. So and yeah, it's kind of performs like I would expect a hack to perform. So it's not great, but it's better than nothing.
yeah, pretty intuitive. Thank you.
We actually did a control here. What happens if we don't do the coupling? So what happens if we just provide the secondary structures for the parent and children and do not couple the processes? And for that, I have a backup slide. So basically, there we found that if we do not couple the two processes, um, we only get a 0.4% success rate. So having this coupling between the processes really increases the success rate of multi-state design by a lot.
Wow. I'm wondering what failure modes you see at this level.
Sorry, could you please repeat the question?
I was asking about the failure mode. I was that
Yes,
you
yes.
see. Yes.
Okay, so basically what we see is basically two things that we get in terms of failure modes here. One of them is we get confident predictions for all states. However, one of the children still has the better sheet, or both children still have the better sheet. So in some way, the structure of the parent won out in the sequence design step, and we get structures where basically the children look like taking the parent structure and just splitting it in half, right? That is one of the failure modes. And we see this also in these partial success cases over here. These almost exclusively look like we have the parent that folds as expected. We have one child that folds as expected, and the other child looks like a cutout of the structure of the parent, right? So, but all of the predictions are confident. The other failure mode is either the parent or the children are not confidently predicted. So
Wow.
yeah, which is the more boring failure mode in the sense that, yeah, we didn't manage to design anything at all, while maybe we can learn something from the failure modes where one or both of the children are predicted to fold like the parent or vice versa.
Wow. Yeah, that's perfect.
All right. So much for kind of general things to do with salad. So after publishing this paper, we've been looking into collaborations to get this actually tested in the lab, and to bring this to bear on real problems in protein design. And to do that, we had to build a bit of a framework around that. This is currently still work in progress, but it powers currently all of our collaborations that work using Salad. And so this library that is currently on GitHub, it's called FlexCraft because it's a Bindcraft inspired library. And what it does is it provides the tools to stick together Salad with AlphaFold, with Protein MPNN, and with a whole bunch of other tools for protein structure prediction and protein design, also Rosetta, for example, to build these end-to-end pipelines. Because in the end, what you need for protein design, it's not only a backbone generator, but you need a whole pipeline that goes from backbone generation through sequence design, structure prediction, all the way to design selection and codon optimization, that you have something that you can actually stick into the order mask at IDT or twist and order as actual DNA to be tested in the lab. And using this library, we're now collaborating with a number of labs to do a bunch of interesting applications. For example, with Lucas Miller's lab, and in particular, Emma Weissert, a student in his lab, we're working on designing very large symmetric protein assemblies using Salad. So we're trying to push the boundaries of what's possible to design with a protein structure diffusion model and go beyond the limit of 1,000 amino acids. The goal in the end is to push this all the way to de novo designed assemblies of 3,000 to 6,000 amino acids in size. Here's a couple example structures that we were working on. And just recently, we were able to get some first experimental confirmation for the smaller designs. These four here. So basically, what our collaborators at Lucas Miller's lab did was basically take the proteins that fell out of the pipeline, um, order them as DNA, express them, purify them, and stick them through size exclusion chromatography to see, well, do they have the right size? Because we designed symmetric assemblies, right? And so we know what size the monomer should run at in size exclusion chromatography, and we know what size the complex should run at. And so we were able to get confirmation that indeed, for a number of these designs, the complex, so the protein ran as the complex at the correct size. And also, the complex was very thermostable. So even after 1 hour of cooking the complex at 95 degrees, it's still mostly eluted at the same size. So these complexes seem to be very stable. Now, further down the line, we hope that we can get actual crystal structure validation for some of these. But for now, those are kind of the first experimental results from this collaboration. Right. Um, and this is not the only collaboration we're working on. There's a number of different things in the pipeline with different labs. Some of them want motif scaffolding. Some of them want symmetric assemblies. Some of them want something else entirely. And we're hoping to get quite a bit more experimental validation for salad over the next couple of months. Right. So, in summary, what did we do? We made salad, a model which is fast and reaches state-of-the-art designability, even when compared to models that were published even after Salad. So looking at Proteina, for example, Salad still reaches state-of-the-art designability across multiple design tasks. It can be adapted to new tasks without retraining. So if you have some very niche, very specific type of protein design task, chances are you can use Salad and the FlexCraft library to write a script, to write a pipeline, which uses Salad to produce backbones and to produce protein sequences for that task. And for the next steps, while we're working on validation with our collaborators, we're also looking to integrate additional modalities into the setup model that can currently not be designed for, which is mostly making salad able to design in the presence of nucleic acids and small molecules, and making it easier to compose multiple models in a single design pipeline. Right. With that, I think I've reached the end of this talk. I'd like to thank you for inviting me. Thank you for your attention. I also thank my lab and all of our collaborators because without them, this wouldn't have been possible. Right. Now I'm
Okay.
open to take your questions.
Thank you, Michael. Thank you very much. Yeah, I have a few questions. And I think the first one is about when we come to think about salad in a way between sort of like a trade-off between longer proteins or proteins
Yes.
with longer amino acids and then flexibility. Because at first you showed that slide where you had that triangle balancing speed and quality.
Yes.
But where does do you think salad like belong? Does it bring out that flexibility component, projecting it to the level of designable or designability in that sense?
So in the end, the trade-off triangle, right? The this thing, right? So I would say salad is very much at the maximum of speed, but it's also the quality is also comparable or better than previous diffusion models. In terms of flexibility, I would say that it is somewhat less flexible than hallucination-based approaches because sequence conditioning is more difficult in salad. I wouldn't say that it's impossible, but it's more difficult because salad operates mostly on structure. So if you want to do sequence conditioning, which is very easy to do with hallucination models, you would have to first run a salad trajectory, then predict a sequence, and then filter based on that sequence. And so you would have to run a whole lot of trajectories and get your designability or your sequence design basically from filtering your salad designs in the end. So I would say it's somewhere over here, if you can see my mouse. So
Yes.
basically maxing out quality and speed, but not quite at the maximum flexibility. Right.
Okay. Um, ah, uh, Maybe this might be the last thing. The concept of hallucination in protein structure design. When it comes to language models, it is understandable for me. And how would you describe hallucination in these diffusion models when it comes to uh,
Right.
protein structure generation? Yes.
Okay. Okay. So this is an interesting question because there is a concept of hallucination that is very different in protein design compared to language models. So with language models, we think of hallucination as something inherently negative, right? So we do
Yes.
not want our language models to hallucinate. But what kind of happened historically in the field of protein design is there was this, I think, 20, 2019. It was a while ago. It was a while ago. There was a preprint from Anishchenko and colleagues that described a new method for protein design using a pre-trained machine learning model. At this point, there was still TR Rosetta, so kind of an improved open source implementation of what was AlphaFold 1 back then, right? And they did this thing where
Yes.
they started from a random amino acid sequence and started optimizing this to maximize TR Rosetta's confidence. And they called this process of optimizing a sequence hallucination for some reason, for whatever reason. Um, and this term has stuck. So usually when someone in the protein design field speaks of hallucination, they mean this optimizing of a protein sequence using a structure predictor. Um,
Wow.
so very different thing from language models.
Wow. Wow. Wow.
However,
Wow.
the concept of hallucination in language models transfers over to basically any type of model you have also in protein design, because these models also produce structures or sequences that are, well, that don't make sense, right? That are not real or that are not realistic enough that they can be made in the lab. And currently, we're kind of stuck with this in the field. So currently, the way the field is trying to guard against this, so trying to filter out unrealistic designs so they never make it to the lab, is by doing this sequence design and structure prediction check here in the design pipeline and maybe adding biophysical features from Rosetta or from molecular dynamics to that. So basically, you get a structure out of a structure generator, and you do not know if it's realistic. And the best thing we can do right now to check if it's realistic is to do sequence design and structure prediction and check if the structure prediction is confident and matches the original design. And then if the structure has reasonable physical properties, like not too much hydrophobicity on the surface, not too much charge on the surface, low predicted aggregation propensity, stuff like that. But none of these features really eliminate bad designs. In the end, the only way you can see if a design was realistic or not is by ordering the DNA and making it in the lab. So yeah.
Brings me to my last question. When we look at salad, it is yes balancing. I hope my internet is good. It's balancing the quality, speed, and then some level of flexibility. But again, designing the structure, how do you mind about the function, like the function of the protein itself that has been created, like function of sort of prediction at the end of the day or evaluation? Like, did you look at that and how does salad fare at that level?
Right. So at the moment, for salad in particular, we do not have functional validation. So we do not have validation of, enzymatic activity or something. However, if you look at the rest of the field, at different approaches that were used, for example, for enzyme design or for inhibitor design, using very similar methods, the way this is done usually is at the level of the structure, right? So if you want to design an enzyme, what you usually do nowadays is you get a theozyme. So you get a theoretical or theoretically optimal active site of the enzyme. Basically, you can get this from uh, quantum mechanics computations. of a transition state of the reaction the enzyme is supposed to catalyze, and then you optimize to this how side chains, so side chain atoms should be placed to stabilize that transition state. So you basically have a structural description of what you need or what you want for your enzymatic function. And then you go back to a motif scaffolding like approach. So you take a thiozyme. Now imagine this motif here is your thiozyme, and you graft this thiozyme into a backbone. And so in the end, it's very similar to what we do here. And in the end, the types of things that are checked before ordering this enzyme are also very similar, because what is usually checked then is if the geometry of the thiozyme, the geometry of this theoretical active site, is matched by the designed protein. And if it does match and then also meet some biophysical parameters, so there we go back into Rosetta and we compute energies, or we might even go into QMMD and compute things there. And if that matches, then things are ordered. But to my knowledge, in these types of structure-based protein design approaches, it is fairly rare that function predictors are used at any stage. Instead, the function is defined on the structure level and yeah, validated on the structure level.
Thank you very much, Michael. Thank you very much indeed. And to the
Thank
members
you
who have
for
been on the
inviting me.
call, it's a pleasure for us. Yes, yeah. So I want to end the call by saying thank you again to Michael and to everyone. We meet in our subsequent session next week. Bye-bye, everyone.